Welcome all. In today's lecture, I will talk about the use of selectable marker genes for screening of transformed and non-transformed bacterial cells. So, as I mentioned before, that bacterial transformation is highly inefficient process. So we need certain mechanism or certain selectable marker genes which we can use to differentiate between transformed and non-transformed bacterial cells. So now we discussed earlier that there are two different types of selectable marker genes. So one is antibiotic resistance genes and the other one is enzyme producing gene. Now we talked about the antibiotic resistance genes that there are certain plasmids, they have these genes. So those plasmids, for instance, if we have antibiotic resistance, if we have ampicillin resistant antibiotic gene in a plasmid, and that plasmid is in a bacterial cell, so those bacterial cells will be resistant to ampicillin. And similarly, we discussed that, for instance, in PSC101 vector plasmid, which was the first generation vector that has only one antibiotic resistance genes. We also talked about PBR322, which is another vector, and that has got two antibiotic resistance gene. One is ampicillin and the other one is tetracycline. So we can use these antibiotic resistance genes as one of the we can use them as selectable marker to differentiate or distinguish between transformed versus non-transformed cells. We also have another type of selectable marker gene, that is enzyme producing gene. For instance, like Z gene that synthesizes beta-galactosidase. So beta-galactosidase is one of the enzyme that is synthesized by like Z gene, and that beta-galactosidase will then act on X-gal substrate, and then it will convert that substrate into a product which is blue in color. So if we have beta-galactosidase in a bacterial cell that is synthesized and that is functional, then these bacterial cells will have the ability to act on the substrate which is present in the agar plate. It will turn that substrate into a blue product. So those cells that are blue in color are those colonies, bacterial colonies that are blue in color. They will be non-transformed, and those that are white in color, they will be transformed. We also, this blue-white colony phenomena is also known as alpha-complementation. So how does it work? In this case, we are using antibiotic resistance gene as one of the selectable marker to screen between transformed and non-transformed bacterial cells. Now, for instance, if we insert our gene of interest in a plasmid, and that plasmid is having ampicillin resistant gene. Now we didn't use that ampicillin resistant gene in order to clone or insert our gene of interest into that gene of antibiotic resistance gene. So we have, for instance, if this is the antibiotic resistant gene, we have to clone our gene of interest somewhere here. We did not disturb this antibiotic resistant gene, so it is like it stays intact. Now what will happen when we do the transformation? So we will get these two different types of bacterial cells. So one of the cell will be a bacterial cell will be, so one type of the bacterial cell will be those that have taken up our gene of interest along with the plasmid, like this one. So that is transformed bacterial cell, and that bacterial cell did not take the plasmid with our gene of interest. So these cells are non-transformed bacterial cells. Now how we differentiate between these two different types of bacterial cells? What will happen if we add ampicillin antibiotic in a culture medium and let these bacterial cells grow? What will happen that this bacterial cell or a colony will grow as plasmids with ampicillin resistant gene is there. So these bacterial cells that has a plasmid having ampicillin resistant gene, they will grow in a bacterial culture with ampicillin antibiotic. So these cells will be transformed cells. However, in this case, the transformation did not take place, and that's the reason the plasmid is not there. So what will happen if we use culture medium with ampicillin antibiotic? These cells, bacterial cells, will not grow as there is no plasmid. So these cells are sensitive to ampicillin antibiotic. So these are non-transformed cells. Now that's one of the way we use antibiotic resistance gene as a selectable marker to differentiate between transformed and non-transformed cells. Now, what will be the situation if we have like a plasmid having two selectable marker, having two antibiotic resistant genes? For instance, if we have a plasmid, for instance, we have PBR322. Now, PBR322 plasmid is having two antibiotic resistance genes. So one is ampicillin resistant gene and the other one is the tetracycline resistant gene. Now, what we are going to do, we clone our gene of interest in tetracycline resistance gene. So what does it mean? It means that we are going to inactivate tetracycline resistant gene. So the plasmid in which we make one antibiotic resistant gene as inactive, those bacterial cells will not show resistance, so they will not be resistant to that particular antibiotic. For instance, in this case, if we are going to insert our gene of interest in tetracycline resistant gene, what will happen that these bacterial cells, if they take up the plasmid through transformation, these bacterial cells will not be resistant to tetracycline. It will be sensitive to tetracycline, so these bacterial cells will die. On the other hand, they will be resistant to ampicillin because we did not interrupt ampicillin resistant gene. The ampicillin resistant gene is working. Now, if we add ampicillin in a growth medium or a culture medium, then these bacterial cells will ultimately grow. So how? For instance, now we have plasmid with ampicillin resistant tetracycline gene, and we put, for instance, this is the plasmid, and here we have tetracycline resistant gene, and here we have ampicillin resistant gene, right? Now we are going to clone our gene of interest here in tetracycline resistant genes. For instance, we use BAMH1 site. If you just recall or your memory from my previous lecture where I discussed different types of plasmids, that we have a PBR plasmid, PBR322 plasmid, and that PBR322 plasmid has a restriction enzyme site in tetracycline resistant gene. So if you use any one of the restriction enzyme site in the tetracycline resistant gene and clone your gene of interest in that region, what will happen that the tetracycline gene will become inactive, while ampicillin resistant gene will still work. Now we have a plasmid in which we inserted our gene of interest in a tetracycline resistant gene, and then we did a transformation. So what will happen in transformation, as you know very well, that the uptake of plasmid carrying our gene of interest by a bacterial cell is transformation, and we mentioned that we do it through heat shock treatment. So for instance, this is a bacterial cell that has taken up the plasmid in which we inserted our gene of interest in a tetracycline resistant gene. And on the other hand, we have another type of bacterial cell, another bacterial cell that did not take the plasmid, any plasmid, so it didn't work in a transformation. So transformation, these bacterial cells are not transformed. So what will happen now if you take these bacterial cells in which ampicillin resistant gene is working, however tetracycline resistant gene is not working, so we have made this tetracycline resistant gene as inactive by inserting our gene of interest. Now what will happen if we use culture medium or a growth medium and add ampicillin antibiotic? So these bacterial cells will grow, is plasmid with ampicillin resistant gene is intact, and that's I mean, so these bacterial cells will be transformed bacterial cells. So we didn't made this ampicillin resistant gene as inactive. We clone our gene of interest in a tetracycline resistant gene. So the tetracycline resistant gene is not working, and that's why these bacterial cells will be sensitive to tetracycline. However, these bacterial cells, they're quite resistant to ampicillin antibiotic because the ampicillin resistant gene in a plasmid is intact. Okay. On the other hand, we do not have any plasmid here, so these bacterial cells are not transformed. What will happen if we use a culture medium or a growth medium and we add ampicillin antibiotic? So these bacterial cells, where we do not have any plasmid, will not grow as there is no plasmid. So these cells are sensitive to ampicillin antibiotic, and that's how and they are known as or they are non-transformed cells. So that's how we use antibiotic resistance gene for screening of transformed and non-transformed bacterial cells. Okay, so the other type of screening is known as BlueWhite colonies. And as I mentioned before that, so we discussed screening based on using the antibiotic resistance gene, and in this case, we use another gene that codes for a enzyme, and the presence of enzyme is detected by appearance of blue colonies in the Petri plate or in the agar plate. So all those colonies that are blue in color, they will be non-transformed, and those colonies that are white in color will be transformed. So we'll see how does it work. Okay, now if you remember, when I was talking about the plasmids in my previous lecture about plasmids, I discussed one of the plasmid, which is PUC18 plasmid. And I talked about this plasmid that this plasmid has one antibiotic resistance gene, and in addition to that, it has another gene which is based on the concept of lycopyran. So what happens in that we have a promoter region, and that promoter region is attached to the operator region. So if you remember, I discussed that the phenomena of regulation in lycopyran works in a way that we have a repressor protein, and that repressor protein binds to the operator region, this one. And when this repressor protein is bind to the operator region, then the promoter is not accessible for the RNA polymerase enzyme to use the promoter sequence. So we need to remove this repressor protein from the operator region. And in this case, we use IPTG. So IPTG is used as to induce the production of protein of interest, to make this promoter available for RNA polymerase so that they can synthesize the gene. And if there is no IPTG, then the repressor protein will bind to the operator region, and the promoter will not be accessible to the RNA polymerase enzyme. So that is very important to detach repressor protein from operator through IPTG, because when IPTG attaches to repressor protein, it brings some conformational changes in the repressor protein, and ultimately repressor protein will detach from the operator region. Then it is followed by MCS, which is multiple cloning site here. As I mentioned before, that what is multiple cloning site? So multiple cloning site is that region where we have sites for many restriction endonucleases. So that is the region which we use for insertion of our gene of interest, because of due to the presence of restriction sites for various restriction endonucleases. And then it is followed by LAC-Z alpha gene. So LAC-Z alpha gene basically codes for the N-terminus are the alpha subunit of beta-galactosidase. So how this system works, the bacterial chromosomal DNA will have one half of the beta-galactosidase gene, and plasmid will have other half of the beta-galactosidase gene. So what happens that if we want functional beta-galactosidase enzyme, then these two, the product of two genes, so one of the N-terminal part of the beta-galactosidase will be ligated to this promoter in a plasmid, that is leg Z alpha. So once this leg Z alpha is synthesized, then we have the other half of, which is C-terminal part of the beta-galactosidase enzyme is the gene for that is located in a bacterial chromosome, and it is also synthesized. So the other half of the beta-galactosidase enzyme will be synthesized from bacterial chromosome. Now when these two, the N-terminal part and the C-terminal part, they combine together. I'm not going into a detail about the structure of beta-galactosidase. I have tried to make it more simple, just to understand how the system works. So then we have a functional beta-galactosidase. Now when we have this functional beta-galactosidase, then it can function properly. And what happens that if we have a Petri plate, and in that Petri plate we have X-Gal substrate. So X-Gal is a substrate, and this beta-galactosidase will use that X-Gal substrate, and then it will convert that into a product which is blue in color. The names for X-Gal are the X-Gal substrate is like known as 5-bromo, 4-chloro, 3-indole B-D-galactopyranoside, and a blue color, which is a product, is 5-5' dibromo, 4-4' dichloro indigo. So that's how the blue color is formed. Now this is, the simplest explanation for this phenomena is that the N-terminal part, which is also known as the alpha subunit of beta-galactosidase, it is synthesized from this plasmid. However, the C-terminal part of the beta-galactosidase is synthesized from bacterial chromosome. So when they combine together, they make a functional beta-galactosidase, and that functional beta-galactosidase will act on X-Gal and will convert it into a product which is blue in color. And that's why we see blue colonies on the Petri plate. For instance, here you can see, so this is the Petri plate, and here you can see that these are blue colonies, these are white colonies, again blue, so many blue colonies. So all those blue colonies show that these bacterial colonies are not transformed, because the beta-galactosidase gene, the alpha portion of the beta-galactosidase gene that is ligated to a promoter in a plasmid, is not interrupted, it is active. For instance, in this case. Now what happens if we use this multiple cloning site? As I mentioned before, that we use this multiple cloning site, and we insert our gene of interest into this multiple cloning site. So what will happen? The leg Z alpha will not be synthesized. Now this gene will not be synthesized. We made this leg Z alpha gene as inactive by inserting our gene of interest into multiple cloning sites. So what will happen? That our gene of interest will be synthesized like this one, and the C-terminal part of beta-galactosidase gene, which is available in the bacterial chromosome, will also synthesize its product, which is a C-terminus portion that is there, but there is no alpha portion, because we have interrupted that like Z alpha gene by inserting our gene of interest. So in this case, where we ligated or inserted our gene of interest into a plasmid at multiple cloning site, in that case there will be no synthesis of alpha portion of beta-galactosidase. So what will happen? the N-terminus alpha portion is not formed, while the C-terminus of beta-galactosidase is formed, so no functional beta-galactosidase is formed. Beta-galactosidase is formed. So what will happen? If there is no active beta-galactosidase, it is not going to act on X-gal substrate, and there will be no product. And that's why we see these white colonies on the petri plate. So it's quite simple. If we want to, if we just interrupt, if we interrupt the leg Z alpha gene by inserting our gene of interest, then it means that the alpha portion of the beta galactosidase, which is beta galactosidase, sorry, which is available or encoded in that leg Z alpha gene in a plasmid, will not be synthesized. So that will ultimately give rise to white colonies. So this blue and white colony, by just visualization, we can say that these colonies are transformed and these colonies are not transformed. This phenomenon is also known as alpha-complementation, the phenomena of blue-white colonies. Thank you very much.